First of all, I would like to thank the organizers for giving me the opportunity to tell you a little bit more about our work on the role of inflammation in vein graft remodeling. And I have to warn you, it's a little bit more basic science directed than clinical science directed, so I hope to get you through to this story. We've been told this morning by several talks that vein graft remodeling, vein graft failure is due to the hyperplasia and accelerated atherosclerosis, and I won't bore you too much with the grade of failure. It's high in patients. But what's important to know is that one of the key triggers is the damage to the vessel wall when we graft this. This is a piece of saphenous vein that we got from our vascular surgeons that was left over, from our thoracic surgeons, leftover fragment when it was used for surgery. And you may notice that there's a lot of endothelial cells already gone just at the moment of grafting. So that's a key issue that might be crucial in inducing the vascular remodeling. So if you look at the vessel wall, then we know that the vascular cells are important in vascular remodeling in the vein graft, the endothelial cells, the smooth muscle cells, and the fibroblasts. But next to that, I would like to convince you in the next couple of minutes that also the cells, various cells of the immune system, the leukocytes, the lymphocytic cells, and also the myeloid cells, the monocytes and mast cells, play a crucial role in vascular remodeling in the vein grafts. So these cells are, to my opinion, cells that are crucial in inducing the remodeling processes. So in this presentation, I would like to convince you that inflammation and the immune system really plays a role in the development of vein graft disease. And to convince you for that, I will make use of a story based on the mouse vein grafting model, briefly introduced by Andrew in his presentation. It was developed by Kimball Xu's group, and we work with this model now for about 15 years. And it's based on the fact that you take from a donor mouse a piece of vena cava, which you then place as an interposition in the carotid artery of the mice. And you have these tricks with these pieces of tubing here, along which you invert the artery so that you can get a real tissue connection for this interposition. And you can see this in place here. It's really positioned here. You see this distension coming up. And if you have a graft in position in a transgenic mice that is atherosclerotic, then you can already see after 4 weeks some lesions being formed looking like an atherosclerotic lesion. And I'll show you more about that in the next slide. And if you look at a MicroCT scan, then you see here the graft in position over here. And if you look at the enlargement, here's the normal carotid artery, and here you see the distended graft in this area. So you can appreciate it's pretty small to work with, but we can mimic many of the features that we also see in the human setting, since we can use all the transgenic mice. And the development of the lesion, as Andrew showed, is quite fast. Within 4 weeks, you have this lesion being formed. If you have a normal wild-type mouse, there's a multiple cellular thick lesion being formed. But if we use a atherosclerotic-prone mice and mice strain, and we give them a high-fat, cholesterol-rich diet, you can appreciate that the lesion formation is strong and massive. And if you look at the features of this lesion, there's a plaque-like structure. And if you look after 4 weeks, there are really features that are linked to end-stage human atherosclerotic lesions, like they were introduced as being a late-stage failure on vein grafts. So to work with this model, we started off to study the role of inflammation. And one of the simplest ways to study inflammation is to go for a potent inhibitor. And we started off with using dexamethasone in our mouse model to see the effects on vein graft thickening. And what we did is we gave mice a oral dose of dexamethasone in the drinking water, and we performed the surgery, and you can appreciate that the lesion, as you can see it in the controls, is quite reduced after 4 weeks of analysis. But what's striking in this story is when we looked at 7 days treatment of dexamethasone and sacrifice after 4 weeks, we could observe the same reduction in lesion size as we could observe with 4 weeks of treatment. So that pointed to us that the initial stage of vein graft remodeling might be very crucially driven by inflammation, and if you could interfere in that area, we may have a long-term gain. So that is crucial for the future lines. Inflammation is a crucial trigger in vascular remodeling based on these findings. And this short-term blocking of the inflammatory response in the early stages might be a crucial effect in long-term treatment. Okay, so then we went on to the next stage of our studies, is how can we specifically identify factors involved in vascular remodeling. And then one of the first factors we touched upon is the chemokine MCP1 or CCL2, another name for the same factor. And that is a factor that is known to stimulate the attraction of inflammatory cells to the vessel wall. And MCP1 is in many vascular remodeling processes in atherosclerosis a first factor found. So our question then was, how is this crucial factor in inflammatory cell attraction involved in vein graft remodeling? And for that we used tricks that were based on gene transfer by using two approaches to inhibit the MCP1 signaling cascade. The first approach that we chose was to systemically overexpress a receptor antagonist of the MCP1 receptor, and that's a mutant form of MCP1 lacking a short part of the receptor binding domain. By that it can bind, but it cannot induce a signal transduction. And the other approach I will touch upon in the next couple of slides will be using the local silencing of the MCP1 receptor CCR2 by a trick called siRNA overexpression. And for that we use lentiviral vectors, as have been shortly touched upon by Andy in his previous presentation. So if we overexpress in the mouse by plasmid injection into the calf muscle a form of MCP1 that is lacking the N-terminal domain, then we can see that the lesion development is strongly reduced in this mutant MCP1 infected graft. Infection with a control antivirus is similar like the non-treated control groups. So blocking the MCP1 lesion results in a profound reduction in lesion thickening, in vein wall thickening, and a gain in luminal area, pointing out that this MCP1 pathway might be very crucial as a driving force in the inflammatory remodeling processes in vein graft remodeling. But what can we do with that in a local setting? And for that we went to a trick where we would like to shut down the expression of the receptor of this MCP1. And the trick is that you overexpress a short hairpin RNA encoding an siRNA that can block the expression by using a lentiviral vector. And this lentiviral vector can be applied locally on the vein graft using a pluronic gel, and it results in an effective shutdown of the expression. So in this cartoon, we have this vein graft position. What do? We use a pluronic gel that is loaded with the lentiviral vectors, pasted along the graft when it's placed in position. And then we look at gene transfer. If you look at a GFP marker gene, then you can appreciate very nicely that throughout the vessel wall the green signal is coming up, so we have transfected the vessel wall by this trick, while the control doesn't give any signal. So what's the effect on vein graft thickening? Of course, there was a reduction, otherwise I wouldn't have shown these slides, of course. A profound reduction similar to what we could see when we overexpressed that inhibitor for MCP1. So local application of a receptor inhibiting gene blocking the inflammatory response leads to a reduction of intimal hyperplasia. So inhibition of MCP1 CCR2 pathway in murine vein grafts reduces vein graft remodeling. And local gene transfer is really a suitable possible approach, like has been presented in the previous talk by Andrew Baker, showing all these other viral approaches. Question now is, what triggers the inflammatory response in vascular remodeling? And for that, we look at the innate immune system. We think that's a crucial factor, and I know that you probably will not be too much aware of the innate immune system. The part of that we zoom in on is the Tolllike receptors, complement factors, and mast cells. These three factors are, in our opinion, crucial factors in triggering the vascular remodeling response. So, to make a complex story quite simple, Tolllike receptors have a complex signaling system. The only thing I would like you to remember is that if you trigger the Tolllike receptors via this route, in the end, pro-inflammatory cytokines will be formed. That's our thought. And how can you trigger the Tolllike receptors? Now, these Tolllike receptors, they are usually part of the innate immune system, and they are involved in the clearance of bacteria, but also damaged and apoptotic cells. So the ligands for this receptor are damage-associated molecular patterns, DAMs, factors that are being produced on tissue damage. So like in normal tissue, a damaged tissue, matrix components, parts of the connective tissue, become free. You can get heparan sulfate, hyaluronic acid, fibronectin fragments, but also heat shock proteins and DNA fragments. All of them can bind to the Tolllike receptor, and they can provoke this inflammatory response. So with this knowledge in the back of our mind, we went to the mouse studies and used a mouse strain that was deficient in Tolllike receptors. And I don't have to tell you too much to show that Tolllike receptor 4 deficiency really has an impact on vein graft thickening in this model. This is a model where we have knockout mice, but if we go for an approach using atherosclerotic mice in which we do the local gene silencing trick, but then for the Tolllike receptor 4 again, we do get similar results as we could see in the knockout mice. And if you zoom in, you can see that the thickening of the vein graft wall is profoundly inhibited if we locally inhibit the Tolllike receptors. Another approach of the innate immune system is the complement system. And as you may know or may have known, complement systems is a complex pathway which can be activated in 3 different ways: the classical pathway, the lectin pathway, and the alternative pathway. Essentially, it leads to the conversion of the complement factor C3 to C3a, and that leads to the activation of several factors, and eventually leading to the activation of the membrane attack complex. So in this system, you can intervene at several levels. You can block the C1 activation using C1 inhibitors. You can block C3 conversion by using covalent factor or IGs. Or you can block the C5 activation by C5a receptor antagonists or C5a overexpression. So that's the tricks that we use in our mouse model, and also in a vein graft model in vivo, or vein graft is not a proper word. What we use is we use saphenous vein segments, and we use them in an extracorporeal circulation system during cabbage operations. So on a parallel pump, this vein graft segment was exposed to a blood circulation during 6 hours, and we could add our C1 inhibitor to this circulation and look at the effects. And then we zoomed in on firstly on the presence of endothelial cell, and if we do not perfuse, you can see that we have in this segment about 80% of the endothelial cells left. If we perfuse in this system, the segment, we have a profound loss of endothelial cells, and if we add the C1 inhibitor, this loss of endothelial cells can be prevented to a certain extent. So there are more endothelial cell presence when this is C1 inhibitor is present. And also look here in this graph, we quantified the presence of the C1 inhibitor. Normally it's present about 5% of the lesion area, but if we added the C1 inhibitor, it comes up. And this is mainly in the intimal, the longitudinal layer, so this layer of the saphenous vein. If you look at the circular layers, these differences are not that big. That's this layer. So with this knowledge, we went back to our mouse model. And what we could see there is that in addition of the C1q inhibitor, that it also protects the endothelial damage in our mouse vein grafts, resulting in a profound reduction in the intimal hyperplasia seen in these vein grafts. So C1q inhibition can be a profound way to block intimal hyperplasia. And then the last part of the story will be zooming on the complement factor 5a, which brings a very interesting aspect to our thoughts. First of all, we could block C5a by using a compound that blocks the receptor, and I can convince you very easily that this blocking has an effect on intima hyperplasia in the vein graft lesions very nicely. Lesion size is reduced, and if we look at the number of cells in the lesions, then we could see that the number of macrophages infiltrating this lesion is profoundly reduced. But most interesting also, the number of mast cells are reduced by 50% approximately. And if we do the experiment in the other way around, we use a control mouse and we give C5a to the outside of the vein graft using the pyronic gel trick again, you can appreciate that the lesion size will grow and there will be a bigger lesion being formed over here. And also compared with that, we do see an increase in inflammatory cells. The macrophage number is significantly increased, and also the mast cell number increases. So how relevant is that? If we look at the mast cell, which is a first reaction for inflammatory triggers, then it can be triggered by several compounds. Bacteria, viruses can trigger the mast cells. IgE is a well-known activator of mast cells. LDL receptor is oxLDL specifically is a crucial factor. But also the complement factors C5 and C5a and b is a crucial triggering factor for the mast cells. So with this knowledge, we went back to our mouse model and we added again the C5a to our vein grafts, but now in presence of an inhibitor for mast cells, cromolyn. And what you can appreciate is that if you add cromolyn, you do see a very slight reduction in vein graft thickening. If we give just C5a, we do see again the increase in lesion size. But if we give C5a in combination with the mast cell inhibitor cromolyn, we do see a strong reduction of this factor. So that points to an approach that C5a induction might be crucial for activating that mast cell as a crucial factor. And mast cell is a cell that can secrete many factors, cytokines, to drive inflammatory processes. And also you can see the same role in pattern in the amount of macrophages in the lesion. So we are convinced at this moment that mast cell activation via complement factor C5a may modulate the atherosclerotic processes in the vein graft remodeling, and that might be a crucial factor to study in more detail. So to conclude, I think that I did a good job to convince you that inflammation is a key triggering factor in vein graft disease, at least in mice, and that inhibition of these inflammatory responses may have a very potential therapeutic, big therapeutic potential in this vein graft disease, especially as you take in mind that the very, very early stages are crucial for long-term effects. So, and I can imagine that by doing this kind of treatments just prior or around the surgical intervention, it might have beneficial effects. And that's where I would like to leave you without forgetting to thank all the people that contributed to this study in our group.